Now here you can see that in this chapter 11, we have to only do the genetic engineering and especially the recombinant DNA technology. Okay, so introduction we had already gone through what is biotechnology. Now here we see that we will be dealing with genetic engineering and recombinant DNA technology. So okay, now what are the basic facts? See, if you can follow your book, the three basic procedure that is followed is: number one, you see, we have to identify the DNA with desirable gene. That means that you want to engineer or you want to modify. Then we have to introduce this identified DNA in the host cell. Now earlier I have already given you notes on specific terms. Okay, so you need them today. And here you see the maintenance of introduction of DNA in the host and transfer of DNA into the progeny. Now that means here the identical DNA at the desirable gene, that you known as a recombinant DNA. Then you need the origin of replication and vector to which you can transfer the gene, and again the maintenance, that means you need DNA cloning and the plasmid, that is extra chromosomal DNA. So this is the basic fact. Okay, so today I'll be dealing with that. Now here you see this in this portion. See, the first the DNA recombinant DNA is required, then the gene cloning, the gene transfer. Okay, so these are the basic techniques that we would. Now here you see. Now see, first is basically what is genetic engineering. Now you see, genetic engineering, it provides the opportunity for variation and formation of unique combination of the genetic setup. That means what? It provides, genetic engineering provides you the opportunity for variation. What is variation? Deviation from the original and a unique combination of the genetic setup. So basically we follow with the hybridization procedure. Now basically what is hybridization procedure? The hybridization procedure basically uses less, that means inclusion and multiplication of undesirable gene along with the desired gene. That means hybridization means you modify the DNA structure. That is why you produce a variation, that means that is deviated from the original. So for that you need to inclusion and multiplication of the undesirable gene along with the desired one. Desired one is your engineered one and the undesirable one, that is basically present in the progeny. Okay, the original form, and desired one is engineered. So you need inclusion, that means you need to introduce this desired one along with the original one and produce a new recombinant variety. Okay, so now what are the techniques involved? The techniques involved basically, they are the recombinant DNA, the gene cloning and gene transfer. So these are the three things that is very, very important. Now what is the recombinant DNA? And here this recombinant DNA provides a main role to it. You see, the piece of DNA selected. Now here you see the piece of DNA selected would be able to multiply itself. That means what? This recombinant DNA is the piece of DNA that will able to multiply itself in the progeny cell. That means progeny is the future cells, okay, of the offspring of the organism. But you see, this is also another part, has to get integrated. What is integrated? Within the genome. What is the genome? Is a gene structure of the recipient and may multiply and be integrated along with the host DNA. That means that what has to be integrated will have the capacity to go along with the DNA. The alien DNA becomes the part of the chromosome. You see, alien DNA, that means the foreign spliced DNA. Okay, this alien DNA becomes the part of the chromosome which has the ability to replicate. That means what? The host DNA will imbibe the alien DNA and will become the part of the chromosome and will have the ability to replicate. That means it has to get introduced in it. And what is the specific DNA sequence? Now you see, specific DNA sequence is a part of the DNA that forms the origin of replication. This origin of replication is also an important technique, and this is responsible for origin of replication. That means until and unless this part of DNA is there, the origin of replication, the gene cannot replicate. So the alien piece of DNA can replicate and multiply itself in the host organism. That means they can make copy of the cloning. First it has to get integrated in the DNA, then secondly it has to go to the origin of replication. That means it has to imbibe in it, then it has to get cloned. That means it has to be made copies. Now the recombinant DNA molecule, you see, it is what is recombinant DNA molecule? If you have to define, see, basically it is an artificial or created DNA. That means you have linking a gene encoding antibiotic resistance with the native plasmid. Plasmid, already I had told you what is plasmid. It is an autonomously replicated circular extra chromosomal DNA. So this part of the DNA, basically you find it in bacteria alone. Okay. Now you see, nextly we have this you see, tools of recombinant technology. So this part we would be dealing in the next class.